We are going to now turn it over to the speakers that we have joining us from Michigan State University. We're gonna, so as I mentioned before, one of our big goals was to promote cross-campus discussions. And so I'm thrilled that we also will have a cross-university discussion by having our colleagues from Michigan State join us. Dr. Gustavo De Los Campos is an associate professor in the Department of Epidemiology and Biostatistics. His background and research interests center on quantitative and statistical genetics with a focus on the analysis and prediction of complex traits and diseases. Dr. Jade Mitchell is associate professor in Biosystems and agricultural engineering. Her research focuses on human health risk analysis, and she's collaborating principal investigator in the Center for Advancing Microbial Risk Assessment, where her work includes characterizing the risks associated with human exposures to pathogenic agents in environmental media, such as air, water, and soil. So please welcome Dr. De Los Campos and Dr. Mitchell.
Hello everyone, and thank you very much for organizing this very nice event and also giving us the opportunity to share some of the work that we have done. This work was done by a group of faculty from the Epidemiology and Biostatistics Department and Biosystems Engineering. The group has been led by Claudia Holzman, and we started to work about 1 year ago to try to contribute or provide technical support for decisions around many COVID-19 related problems in campus. Today we are going to feature two pieces of work that we did. One is related to the evaluation of an early detection program based on pool testing using saliva samples. And the other one that Jade will talk about is about the development and implementation of a monitoring system based on wastewater. So a little bit of background. In the fall 2020, so I apologize for the typo there, MSU developed an early detection program for screening that used pool testing with saliva samples of saliva. And the idea of this program was to be able to develop a screening program that can test large number of individuals. And the program was successfully implemented in the fall 2020 with a relatively quick turnaround, where results are returned within 24 to 48 hours. And then towards the end of the fall 2020, the administration wanted to know whether there was merit for expanding this program and also for making it mandatory for some groups, those living in campus and attending in-person classes. So the goal for our group in this contribution was to try to estimate the potential impact or campus impact of expanding and making mandatory for some groups the early detection program. So for that, we were working already with stochastic simulations for prediction and forecasting of disease development. And basically what we did, our model is essentially a modification of the standard SIR model, susceptible, infected, recover. Where now in this version, we also have compartments for isolation of people that receive a positive test from this screening program. And because we have some numbers of false positives, we have an isolation compartment for false positives that after the isolation period return to the susceptible compartment, and an isolation compartment for those that are infected that after that they return to recovered. In this modeling scheme that we use, we track people individually daily. Contacts are a network of contacts is generated every day. People also have a schedule for submission of samples to the system, and they receive the results with a lag of 2 days. And unlike the standard SIR model, we have vectors of probability that control the likelihood or the probability of transmission when a contact takes place. Some information about the parameters. We assume a prevalence of 15 over 1,000. This was towards the end of the fall, and we were trying to forecast for the spring 2021. We assume 6% of people were already recovered. In the model, we have a vector of transmission probability. So when a contact between an infected and a susceptible person is realized or happens, the probability of transmission vary between near zero probability at day 1 of infection to about 30% day 5 to 7, and going down after that. The test specificity, based on the data we have, we assume it was very high, and the test sensitivity also varied by day of infection, from almost 0 at day 1, with a peak of 95% sensitivity between days 4 to 7. We considered 3 scenarios in terms of participation. The baseline scenario where there was no early detection program, and then 2 scenarios, one with 50% of the people participating in the program and 100%. This one was unrealistic, but we just wanted to have the whole spectrum of potential for the program. We assume that people that receive isolation recommendations, they comply with 80% probability. As Marisa mentioned, there are a lot of parameters and uncertainty, and so one needs to make assumptions about that. And so in that case, we assume 80% compliance. In terms of the contacts, like I said, we generate a network of contacts every day at random from a Poisson distribution for every individual, and we had 2 parameters, 2 possible values or scenarios for the lambda parameter of the Poisson, 0.55 and 0.6. The R0 in our model is implicitly determined by this parameter and the vector of transmission probabilities. So some results, and I'm going to focus only on a few of these curves. What we have here is the expected number of cases over a semester, roughly 120 days. For the here on the left, for lambda 0.55, which is a lower R0, and lambda 0.6, which gives a higher R0, with 100% participation. Sorry, let me go back. Forward, sorry. With 100% participation and 50% participation. In each of these plots, we also have the baseline, which was 0 participation, here as a reference, right? So what we saw from the simulations is that we were, for this lambda parameter, we were forecasting about 1,700 cases for a population of 10,000. All these simulations are for 10,000, or with the higher value of lambda, about 2,600 cases. And what happens with the implementation of the early detection program, and I'm going to focus here on the blue curve, which is for weekly testing, so people submitting samples every week. What we saw is with 100% participation, almost a 70% reduction in the expected number of cases. And with a more realistic scenario, with 50% of the people participating, about a 40% reduction in the number of cases relative to the baseline. In relative terms, the patterns are similar for the higher R0, but of course, in numbers, the number of cases that are expected are much higher with the lambda, with the higher lambda. So based on these results, we recommended to implement the early detection program with weekly testing, and that program was implemented. Well, before I move into some details about the actual implementation, these stochastic simulations also allows us to produce confidence bands, as it was pointed out before. And so here are 80% confidence bands for the baseline scenario and for the weekly testing setting. The program was implemented. As of today, it has conducted more than 130,000 screening tests with about 13,000 participants involved. And if you are interested in data from the program, it is available in the MSU's dashboard. And we are currently analyzing data that the program has generated, which is very interesting and is allowing us to learn a lot about the population in the MSU campus. We also launched a survey aiming at characterizing people's attitudes towards vaccine. And the next step that we are currently working on is evaluation of the fall 2021 and trying to evaluate where there is merit to maintain the program under different scenarios regarding the proportion of people that are vaccinated. So this is where for the detection program, I'm going to now render the floor to Jade, who is going to tell us about the, let me see if I can go back, Jade. There you go. No, this is, there you go. Sorry for the technical. So Jade is going to talk about the wastewater monitoring system.
Thank you, Gustavo, and thank you to the organizers. I appreciate the opportunity to speak here today. Like most of the other speakers, this isn't my typical research program. It's usually microbial risk assessment, but wastewater-based epidemiology has been growing, especially over the course of the pandemic, in the areas of analytical techniques and discussions about standardizations of approaches. But we're starting to catch up with the data analysis and the modeling. So I'm going to talk about what we've done on campus so far and how we've assessed our wastewater program. Dr. Joan Rose and her lab started collecting samples in May of 2020. These were grab samples at 4 different sites. And overall, we found that we were seeing higher concentrations of the wastewater signal with increases of cases in the community and even at the building level when we were able to isolate buildings. So we operationalized this wastewater surveillance project in the fall along with the EDP, and then later expanded it in the spring when we also had mandatory testing within the EDP, and that provided us a richer data set for modeling. When we expanded, we installed automatic samplers. So to replace those grab samples, we're now able to collect excreted waste over a larger period of time, which would represent more of the residents of each building. We looked at 6 different dorms specifically that could be isolated, in addition to a couple other locations off campus. We sampled twice weekly on Mondays and Wednesdays, mainly because of the time that it takes to run the analysis, and we wanted to have a weekly report for Friday. We analyzed 2 gene targets, but we have since expanded this to also look at variants. And the data that we have for comparison, or that I'll show today, is the isolation counts, which we have the most confidence in within each building. Isolations are based on either confirmed clinical samples or confirmed saliva samples. We also have the populations in each building, which are updated weekly, but we're pretty steady over the time period. So this is what our data looks like. At this scale, it's difficult to see the trends, but we pretty much had a good deal of agreement with the wastewater signals and the number of people that were isolated in each building. What we wanted to look at was the autocorrelation of this data. So in the literature and what we had previously seen with the June outbreak in East Lansing is that wastewater could potentially give us an early warning. When we did our analysis on campus, we actually found that we had the highest correlation with a 3-day lag, though you can see that the data clusters around that 3-day lag point that I have here with the red line. So it's anywhere from a 5-day lag to a 2-day warning. And we really think that this is really just an artifact of when we're sampling and when we're analyzing the data, since we don't have weekend cases. So next, we plan to do a Poisson regression, and we use that 3-day lag in that Poisson regression. Now, there are several approaches that emerged to model this data, especially mechanistically. I really appreciated Marissa's comments about how scale matters. So the models that have emerged are mainly at the wastewater treatment plant level. And there we have lots of mixing and a high residence time, but by design, buildings are, made to flush sewage away as quickly as possible. So there's some uncertainty in the model form and which parameters are really needed to model building scale level wastewater signals. Oops, I was gonna have you go back, but that's okay. This slide went a little bit faster than me. The next thing that we looked at was doing a Poisson regression in order to evaluate the predictability of our wastewater signal or concentrations with the isolations as our dependent variable. And we did this for the 3-day lag and without the lag at all, since it isn't consistent over time. We fit a mixed effects model so that we could look at differences or random effects related to each of the dorms. So overall, we found that our wastewater signal was significant with and without the autocorrelation corrector, which is actually promising. We did see that when we look at our random effects, though, that there are some differences in location. And we would expect this, especially related to the wastewater composition. So buildings that have a cafeteria or have a large laundry facility will have a different composition than other buildings. So I know there's a lot on this slide, even for a modeling symposium, but if we break apart our wastewater concentration, it's really the result of the population that's shedding, their shedding rate over the total population and the amount of wastewater that they're using. We can estimate constants for those for now, though we know that they're variable over time. And that would allow us to simplify our understanding of isolations just based on concentration times the population. So that's what we did to determine if population was a necessary correction or useful correction factor. Unfortunately, because of the limited data just over the single semester, our mixed effects model didn't converge here. So we fit a fixed effects model just on the time lag data to look at predictability of the wastewater signal versus predictability of the wastewater signal and the population correction. And here we actually found that normalization with the building population didn't really improve our model. So this again is good news for moving on to other buildings and scaling up, though we know that there are many different ways of normalizing that might also need to be considered. So what we found is that the wastewater surveillance in conjunction with the early detection program seems to be a viable option. We have good predictability. It's a viable option for scaling up and may actually help us do more targeted testing, since broad testing using the saliva method is very resource intensive. We also want to evaluate other mechanistic parameters and model forms, including flow, which we can estimate with water usage in each building as a surrogate. And then looking at a fully mechanistic model that would include stochastic variables for shedding rates and excretion, since we know that those are variable over time and based on the point of the disease, in the disease process, I mean. And then finally, we'll be looking at the predictability of our wastewater signal for variance. So with that, I'd like to thank our lab staff and our sampling team. I've got acknowledgements on the next slide, because this is a huge effort to do all the sampling and analyze the data within a day for reporting. Thank you.
Thank you very much. I have a question for you, but because I'm moderating the panel, I think I'm going to save it for the panel time.
Okay.
So excellent. Thank you so much for this great presentation.